This is Elisa Schwenk, and today I'm going to talk about the prokaryotic replication fork. So, first, let's begin with the circular prokaryotic chromosome. Before replication can begin, an origin of replication needs to be established. Once the origin of replication is established, replication can begin in both directions, resulting in two replication forks. These replication forks are what we will be focusing on today, so let's zoom in on the right one. If we zoom in, we can see the DNA begin to separate and unwind. DNA is constructed by base pairs made of nucleotides that are bonded together by hydrogen bonds. These base pairs are further elongated by phosphodiester bonds, which allow enzymes to add more nucleotides to a growing strand. Next, let's look at the ends of the strands. As you can see, the strand beginning in the top left is labeled 3', with the other end being 5'. The bottom is complementary to the top. Since nucleotides can only be added to the 3' end, the leading strand will continuously replicate DNA, and the lagging strand will replicate DNA in chunks. Before replication can begin, the DNA strand needs to be unwound, and the hydrogen bonds between base pairs need to be broken. In order for the DNA to unwind, a topoisomerase called gyrase relieves the supercoiling ahead of the replication fork. Next, helicase attaches to the replication fork and, in this example, moves right, breaking the hydrogen bonds in the process. While this is occurring, single-stranded binding proteins begin binding to the single-stranded DNA. These proteins protect the DNA, stabilize it, and prevent hairpins from forming. Now that the DNA is unwound, replication can begin. First, let's look at the leading strand. DNA polymerase III binds to the leading strand and begins adding nucleotides to the 3' end. This means DNA is added in a 5' to 3' fashion. The DNA polymerase III continues to move down the strand, adding nucleotides to the 3' end. Next, let's look at the lagging strand. Since nucleotides can only be added to the 3' end, DNA cannot be synthesized continuously along the lagging strand. First, a primase needs to be added. This primase creates short RNA primers for DNA polymerase to follow by adding nucleotides with an open 3' end to the lagging strand. Once the primers are added, DNA polymerase III can now begin adding nucleotides to the 3' end of the lagging strand. This is done along the whole length of the lagging strand, creating Okazaki fragments, which are short sequences of DNA nucleotides which are synthesized discontinuously and later linked together. Once DNA polymerase III has sufficiently elongated the new strands, DNA polymerase I binds to the beginning of the primers and continue to move along the primer, removing the RNA primer and adding nucleotides until the end of the primer. Once this is complete, ligase comes in and attaches the fragments together. After this is complete, you'll have a continuous strand of replicated DNA on both sides of the original parent DNA.